Hi everyone, in this podcast we are going to take a look at enzymes. The objective, identify the purpose enzymes serve in biology, and identify factors that can affect the rate of enzyme activity. First we need to go over a few things. So what is an enzyme? Okay, this kind of is in the wrong spot. Let's move this down. Okay. An enzyme, a specific if you hear anyone in the background, that's my daughter screaming at the cat. All right. An enzyme, a specific type of protein that serves as a biological catalyst. This means that it speeds up the chemical reactions that support life. Some reactions digest or decompose substances, and some synthesize, therefore they build things. Slide this down. Now, without them, all of the chemical reactions that make up your metabolism would fail. Now, a misnomer. The word metabolism does not just mean how well your body makes energy from foods and sugars. It means the sum of all of your chemical processes. That includes how well your body uses insulin and so forth. Okay, here I have listed important terms. We have substrate. Now these are the terms that are going to be used later on in the slide, so please pay particular attention. Even consider going back to them and taking notes on them. You could pause this at any time that you need to. Well, substrate. Substrate, well, they are the reactant or reactants the enzyme is affecting, while the product is what is being formed, so the end result. The active site is the location of the enzyme that attaches to the substrate. Now the enzyme-substrate complex, when the enzyme and the substrate are locked together, that's called the enzyme-substrate complex. While the lock and key is a descriptive term to suggest that the shape of every enzyme's active site is specific to the shape of every substrate. Move this up some. Now, enzyme activity. That's the speed at which the enzyme makes its products. When it is at its best, we call it optimal or optimum rate. We have pH. pH is a measure that identifies a fluid as acidic, neutral, or basic. Denature. When an enzyme shape is altered. When it's altered, the enzyme will not work. It's optimum. And concentration. Concentration is a term that discusses a solution. We have solutes dissolving into solvents. So this relates to the amount of solute dissolved in a solute. Solute dissolved in a solvent. Our most perfect example is salt water. Salt dissolves into water. And lastly, we have ace. Now this is a suffix. A prefix goes before a word to modify its meaning. Suffix goes at the end. Typically when we have words that end in ase, you have to be suspicious that is an enzyme. So we say it's the suffix used that usually identifies a specific protein as an enzyme. So what does this actually look like? Right here we have an enzyme drawn more like a cartoon and a substrate, and they fit together. We call this the lock and key model. The enzyme and substrate lock together, perfect fit, they work together. But down below is a real representation. This is a protein, and look at its very unique shape. Here is a substrate, and the substrate is now located inside the active site and the enzyme works. Let's take a look at a full representation, more like the cartoon model, but it does describe it quite nicely. Over here, we have an enzyme. That enzyme has a very uniquely shaped active site, and it will only work on sucrose, a disaccharide. Now the sucrose here is now located inside the active site. Follow along, and we notice that the sucrose has now been broken. A chemical reaction has occurred, making a glucose and a fructose. They have been released to the environment, and now they are free to do whatever they need to do. What's interesting though is take a look at the enzyme. It is now unharmed, free to work once again. Now in this slide we are showing you two ways enzymes can work. Again, we're using the cartoon version of our enzymes, and in this one right here we see we have substrate, substrate, enzyme, enzyme substrate complex. When it's done working, we have a product. This is a synthesis. Now, on the other hand, let me erase these. Bear with me. There we go. Shrink this one. Take a look at this one. Make it bigger. Now in this picture, we have substrate, substrate, enzyme, lock and key model. Active site is right here. Now the enzyme does its work and it produces two products that were once part of the reactant, the substrate, and well, this is a decomposition or digestion. Enzyme is unaltered, la-di-da, wonderful thing. Now, the bad news. Here we have a picture of a protein. It is an active, functioning protein. Let's call it an enzyme. Sometimes bad things can happen to enzymes. Either the pH of the environment that protein is in is not correct for that protein, or the temperature is too high. What that will do is that will unfold or sometimes even crack that protein into different pieces. And the active site, if this were the active site, notice it doesn't exist anymore. That enzyme is broken, it's dead, it cannot be used. Okay, so I mentioned pH. Here is a pH scale. It ranges from 0 to 14. Sometimes the scale is written from left to right. It doesn't really matter. Neutral is 7. Well, as you get lower, it becomes acidic. Now, the further you get away from neutral, the stronger the acid becomes. On the other side, getting higher than 7, that's where our bases live. The further you get away from 7, the stronger the base becomes. Let's take a look at some examples. Pure water, 7. Milk, slightly lower. Black coffee, I love this stuff. It's acidic. Tomato juice, obviously, tastes sour. It's acidic. Orange juice, vinegar, lemon juice. Battery acid. Do me a favor, please don't ever drink battery acid. Bad idea. Now, movies have you believe that acids are more of a problem than bases. I suggest the opposite. Take a look here. Seawater, base. Baking soda, base. Fine stuff. Milk of magnesia, helps your tummy, base. Ammonium solution, base. Soapy water, everything that feels slippery, even bleaches, are bases. Bleach, oven cleaner, a liquid dry cleaner, all bases. Now the reason why I suggested the opposite and bases are worse is because you would not assume that they could hurt you more. They could hurt you just as much as acids. Please be careful. Next slide will show you some other examples of common items. Okay, Coca-Cola, acidic. Water, about 7. Easy Off oven cleaner. It's a base, very strong base. Alright, now let's take a look at what this means. Here on this axis of this graph we have pH, and here are the values 1, 2, 5, 7, and rate of reaction. Again, that means speed. The very top would be the optimal. Okay, that's nice. Now here's pH. The green line, if we are at a pH, or the enzyme is in an environment where the pH is about 1, well, that enzyme is not going to be working at its best. If the pH is roughly, let's just say 3, again, won't be working its best. But where would the enzyme be working its best if we're talking about the green line? Right here, follow the line down, roughly 2. Place in the human body where you have a pH of 2, stomach. Right? Now the red line, follow the line down, pH slightly under 7, slightly acidic. Where would the enzyme work its best? I suggest in your blood. But the big question is what's happening here? What's happening here? If we went back to the other slide, we'd have to say the enzyme is denatured. Meaning the shape has changed, therefore the active site has changed, therefore it's not working its best. Next slide we'll talk about temperature. So now let's take a look at this graph. On our x-axis we have labeled temperature. There it is. In degrees Celsius, not to be confused with Fahrenheit. On our y-axis we have increasing enzyme activity. Now what this tells us is as we increase our temperature, the enzyme is working better until it reaches the best, which is about 40 degrees Celsius. It's optimal. After which the enzyme is not working as well or as fast. So what's happening in this range? Well, the enzymes and substrates are both moving faster, so they're finding each other more frequently in the correct way. On this side, though, the enzyme itself has been hurt by the heat. It is denatured and broken. Alright, now this graph suggests that as the substrate, so the thing the enzyme is working on, increases, the enzyme will increase its ability to work until it reaches a maximum output. The analogy that I give in class is this. Imagine there are 10 teachers and 100 papers to grade. The teachers can work only so fast. They'd be pumping out a paper maybe one every 2 minutes. If you keep on giving them more papers, they can only work so fast. So they'll just keep on churning them out until all of the papers are graded. And if we were to complete this line here, eventually it would drop as all the substrate is used up. Alright, so as you increase the amount of substrate, the enzyme will work better until it's working at its maximum rate. It will plateau, then eventually drop when there's no more substrate. So, next slide. Let's try to apply it to just one thought. Alright, so the question is this. Are fevers good or bad? I suggest they're good. Your body's doing it for a reason. What it's doing is it's raising your body temperature, therefore affecting the enzymes of the microbes. The bacteria of your body, they have enzymes in order to work. So, as you increase the temperature of your body, you will increase the temperature of their enzymes and denature them. These microbes, if you denature their enzymes, they're certainly not going to survive. While you, a multicellular organism, tons, millions, billions, trillions of enzymes, you can survive it. Where a fever becomes bad is when it becomes too high and it actually destroys your tissue. That's when you need to be concerned. But please be mindful and pay attention to commercials when you hear them from now on about painkillers that decrease temperature. They are written in such a way to try to convince you, quietly convince you, that all fevers are bad and you should reduce them. In fact, if you do that, you're going to be taking away one of your body's defenses against microbes. If you can handle the fever, in all honesty, you should leave it. Thank you for listening, and I hope this helped. Have a great day.